Welcome to this Microwarriors special edition. I'm Shane Starling, the Senior Editor at Nutri-Ingredients. Today we're going to discuss probiotics, the human microbiome, and their relation to health, wellness and disease across the world. To do that, I'm pleased to be joined by two eminent experts in the area. That's Dr. Mary Ellen Sanders, who is the founder of Dairy and Food Culture Technologies in Denver, Colorado, and Dr. Grigor Reed, who is the chair of the Human Microbiome and Probiotics Centre at Lawson and Weston in Ontario, Canada. So we have the introductions out of the way. Let's sit down and discuss probiotics. So welcome along, Grigor. First question I have for you is, so what is a human microbiome?
That's a great question. You know, when I teach students, I love to tell them that when we look back in history, the only thing living in the world was microbes, and we've evolved from them. And so what is a human and what do we need? Well, we need food, we need water, we need air, and we need bacteria. And we can't live without them. So incredibly, as we've evolved with the microbes. And when I say microorganisms, bacteria, viruses, fungi, other types of organisms. And they're part of us. And for the longest time, people have said, well, they must be bad. We have to kill them all. So then I look back in history and say, well, why did we kill them? So what was it that killed early humans? And obviously accidents and disease. Disease was a very big part of it. And of course, serpents and lions and things like that. And the disease part was the infectious organisms. And through time, we've sort of concentrated more on them, rightly so, because they killed us. And we've sort of done it, though, in a way that's forgotten that most of our organisms are beneficial organisms. And so recently, I'd say in the last 10 years, primarily, because of some large grants in the United States and in Europe, they've started to look at, okay, well, what are the microbes that are in our body? And the microbiome are all of these and all the things that they do. So when someone says the human microbiome, they're really talking about all the organisms in a human. Now, that goes against what we thought, let's kill the organisms, let's kill the bacteria. And we've, as I said, it goes back to because there was certain ones that killed us, but it's led to antibiotics and antiseptics and this whole kind of philosophy that microbes need to be killed. Well, that doesn't make sense at all because most of them are beneficial. So the exciting thing about the microbiome, we used to have to culture bacteria. So my daughter had a school project and she would go to the carpet and go to the toilet with a little swab and she'd put it on an agar plate. And after a couple of days, it grows. And that says, here's the organism that's living. Well, for a long time, we've relied on that. And the new technology, high throughput sequencing, has said, we don't need to do that anymore. We can just look at the sample, take the DNA, and it will tell us what's there. So, for example, in the bladder, we always say, well, the bladder's sterile, and it's only got bacteria in it when you have a urinary tract infection. No. In fact, there could be several hundred bacteria types in the bladder, but they're not doing any harm. Of course, the next question is, why are they there and what are they doing? So the Human Microbiome Project has tried to go to different sites and say, okay, what's in the mouth? What's in the gut? What's in the feces? What's on the skin? And some of it has made the newspapers, for example, that the keyboards that you type, the bacteria on your fingers go onto the keyboards. And so when you sample the keyboards, they said, well, that's Gregor's keyboard because it's got his bacteria on it. So the microbiome has opened up this whole new exciting area of science because, as I say, we used to think things were sterile. Well, if you look at the Nobel Prize in the stomach, they thought the stomach was sterile. And of course, they discovered Helicobacter pylori. And so when I tell the students that we've evolved from the microbes, the next question is, well, how do we get them? And so you say, well, when you're born. And in fact, possibly before you're born. Because you imagine I'm a bacterium and I've evolved a new species, the human species. The species that this human is coming into a microbial world, right? And you have to prepare it for this because many of these organisms potentially could kill you, right? So we think that even the uterus is probably not sterile and that bacteria or their byproducts are getting exposed. So they're priming the baby to say, you're coming into the microbial world. Now, the best priming happens at birth, where they come through the vaginal canal, which has lots of bacteria, and then some of the feces contaminates the baby, and the baby comes into a microbial world. So that's great. Then you say, well, okay, what happens if I have a cesarean section? Well, you don't get exposed to that. And in fact, my twins came by cesarean because they were premature. And so the surgeon opens up and brings out the baby. And often we give them antibiotics, we give them intravenous lines, we give them breathing tubes, and that's a completely different exposure to the organisms. Then the question is that a good thing or a bad thing? And how should we compensate for that? We don't know the answers yet, but that's why the microbiome is so exciting, because we can now look at all the organisms in the mum, all the organisms in the baby, and what it's been exposed to. And the studies have suggested that, for example, if you live in a house with 2 dogs, you'll have less chance of allergy because you're getting the microorganisms from the dogs. But that's another thing. So then you say, okay, baby's born, cesarean or vaginal, what about breastfeeding? And people had known that there was bacteria in the milk, but they assumed that it was skin contamination through the nipple, right? And some researchers in Spain have suggested that maybe it's more complex than that, that when the baby's, just before the baby's born, there's cells in the gut actually pick out some organisms and bring them through the bloodstream to the mammary gland in order for mum to pass on her microbiome to the baby. So that's very intriguing. We looked at the breast itself because people had thought, apart from the milk ducts, it must be sterile. I mean, it's a tissue. Why would there be bacteria there? And we started looking at cancer patients, and lo and behold, we've shown that, in fact, there are microbes in the breast. Now, the ones that we found are things like Pseudomonas. Now, you would think people talk about Pseudomonas and they talk about cystic fibrosis, because that's an organism that is not, it can kill you, this organism. Why is it in the breast? Why is it not infecting the breast? Why is it just there? And is it because it's to be passed on to the mum? Well, we don't know. So the thing that you do is let's study the microbiome of the breast. Let's study it in women who have cancer, those who don't have cancer, those who lactate, those who don't lactate, and get an understanding of why they're there, what they're doing, and what their role is in passing on to baby. Now, when a baby's born, I'm kind of going into babies, but I think this is really relevant. The first few years of life in a baby are amazing because they program us for what happens next. And we're going to talk about probiotics and why you have to keep taking them. And it's because they don't colonize, they don't stay inside us. And why is that? Well, it's because the stadium gets filled up. And the first few years of life, the organisms go into the baby and they form these sort of dense biofilms, we call them. You can imagine lots and lots of them. And they do things like they form a proper intestinal tract through creating blood vessels. They form your immune system. And after a couple of years, it's very difficult to change that. So if you give a probiotic, which is a beneficial organism, in other words, we're trying to restore more beneficial organisms, to give it in the first few years of life, they may stay longer. But once you get to an adult, it's very difficult because I always imagine the University of Michigan Stadium and there's 110,000 people and I go in and say, I want a seat. And they say, no, we're playing our Michigan and we can't give you a seat. So it's very hard to just have them go in and stay. So early part in life is really critical, and the Microbiome Project is giving us insight. And there's some wonderful work in Africa where they're looking at people who've never been exposed to chemicals, drugs, vaccines, anything like that. And they're saying, well, what's their microbiome? And is that the original human microbiome? Because, as you know, we've introduced drugs and chemicals through life, and we've probably changed it. I remember when I was raised in Scotland, we didn't have a fridge. And of course, people would say that's because Scotland's always cold. Well, we know that's not the case. But so we ate lots of fresh food. We didn't eat processed food. We ate probably fermented food more. And I bet you our microbiome was slightly different from what we do now. And so then that's another exciting area, because what does food do to your microbiome? Well, absolutely it does something. And people are now tracking obesity. I mean, why is in America and Canada such high rates of obesity? It has to be the diet. It has to be what we have done with the diet and the microbes, because the microbes are digesting our food. They're deciding, is it going into fat, is it going into the bloodstream? And so the diet becomes really critical. How does the diet affect the microbiome? How does the microbiome then affect the person? So it's really the most exciting thing in science, I think, in the last decade at least.
So Mary Ellen, tell me, is it possible to define a healthy microbiome?
Well, there's been a wealth of information that's been gathered on the human microbiome projects in the United States, in Canada, in Europe, China, a variety of countries. And what's been interesting is, as they've tried to look at how to define a healthy microbiome, they've found that it's really not going to be easy to define it based on composition. So they haven't been able to say, here's one ideal composition of microbes that seems to work for everybody. What they have found, interestingly, is that there is a fair conservation of function among healthy microbiome patterns. And so it may end up being that it's the function, or what these microbes do, that is more important than actually which ones are there.
Mm-hmm. Can you define an unhealthy human microbiome?
Yes, that has been done with studies that have where they've compared healthy controls to controls that have certain disease states or conditions that are somewhat abnormal. And what they've found is yes, the microbiota of people with certain diseases, in fact, is very different from healthy controls. And that's been the case with conditions such as diabetes, allergy, antibiotic-associated diarrhea, autism, obesity, inflammatory bowel disease, a variety of conditions that, in fact, are associated with what you might call a dysbiotic microbiota. So it seems clear that microbes are being disrupted in these cases, but what we don't know is whether or not the disruption is causal or if it's a result of. And therefore, right now, the best we have is an association between the microbes, that changes in the microbiota, and the condition.
Mm-hmm. And this is a big problem in the research, isn't it? In that there's a lot of evidence that shows association, but much less that shows causality. Mm-hmm. So how can association become causality?
Well, that's where intervention trials come in, where you actually do something to try to change the microbiota to have a different composition, and then check to see whether or not you have a concomitant change in the disease state. And that's going to require a very substantial investment in research, and it's going to require a substantial investment in human studies to actually determine, based on interventions that are defined as in that they can have an impact on the microbiota and shift the microbiota, and find out whether or not those interventions can ultimately lead to a change in the disease state.
Is the momentum there to conduct that research? The investment, the relevant bodies, are they getting behind this?
I would hope so. I can't speak globally to that. I'm not sure what kind of research programs are really underway. There certainly are some roadblocks to doing some of those types of studies, but I think that some of them will get done, and they'll get done over time, I believe. I'm not sure how quick that path will be.
What's more important? Is it composition or is it function?
Well, as I mentioned, the function of these microorganisms is really being found to be more important, I think, than the composition. So one of the stories I remember a speaker telling at a conference I was at was comparing the grassy plains in Australia versus the grassy plains in North America. And he said, you look at those grassy plains and they look very much the same. But if you compare the species of grasses in those two different places, they're entirely different. Yet clearly they function in a very similar way. And I think that that might be what's going on in our bodies with the microbes, that the microbes have to have certain core functions, but which microorganism does it maybe isn't as important.
Mm-hmm. Is there a great variety in different microbiomes between individuals?
Yes, you and I would have slightly different microbiomes.
But in many cases, they do the same things. So we call that function. So the function is similar. And there isn't one healthy microbiome or this is the best. And in fact, which one would you want anyway? Would you want Einstein? Would you want Pele to be a great soccer player? I mean, you can't really look at it like that. The thing that amazes me is, if you look at women, and we've studied the vaginal microbiome of women, and some other colleagues have as well, lactobacilli is the organism that is the predominant organism. And you'll find it in Japan, you'll find it in Africa, you'll find it in Brazil, you'll find it in America. And so there's a universality that says, for humans, in that niche, these organisms are really important, no matter where you live. And in the gut, I think we'll find the same. There'll be a commonality. Now, it doesn't, as I said, doesn't mean they're exact the same, but the functions will probably be quite similar.
Is it fair to say that in the Western world, for instance, most microbiomes are dysfunctional?
Yeah, I don't like the term dysfunctional because that's implying that we fully understand what function is and what dysfunction is.
And many of us would say, well, I'm healthy, so how could I possibly have a dysfunctional one? To answer critical questions like that, you need to do the studies. And as I say, the US and Europe, particularly to some degree Canada and Australia, they've provided funding for these studies. Now, the funding is sort of waning, and it's almost as if, well, you've done it and we don't need any more. It's not the case. This is exactly why we need more studies to answer questions like that, because you don't know the answer, and it could be critical to not only quality of life, but longevity and the types of conditions that we develop.
So in terms of the health benefits then, we have, I guess, the most researched areas are gut health and immune health, but what else exists in the research area?
Well, what you're describing with immunity and gut health are really the two broad categories of benefits that are commonly associated with probiotics. But the specific list is actually quite more extensive than just those two. Under the umbrella of gut health, there are endpoints that include symptoms associated with irritable bowel syndrome, antibiotic-associated diarrhea reduction, helping to decrease the recurrence of ulcerative colitis, helping decrease the incidence of necrotizing enterocolitis in preterm infants. So there's a variety of endpoints within there with evidence for specific studies. In addition to that, there's evidence for some types of endpoints that I like to almost think of as having even a broader relevance almost to the public health arena, which would include, for example, decreasing the incidence and duration of respiratory tract infections, of common upper respiratory tract infections, such as the common cold and flu-like symptoms. Which you could imagine, even if the magnitude of the effect is somewhat small, if you were able to have that impact spread across a population, that it could really have a very big impact on public health. Interestingly, one of the studies that was conducted on upper respiratory tract infections, well, not just one, several of the studies that were conducted on the incidence of respiratory tract infections also showed decreases in antibiotic prescription use, which, again, on a public health arena, that's an extremely important observation. So if just by administering a probiotic, you can not only decrease the number of common respiratory tract infections people get, but also decrease the number of antibiotic prescriptions, that could be very important.
Is there any doubt that probiotics can influence the human microbiome?
I don't think there's any doubt that probiotics can influence the microbiome.
So probiotics, people would say, well, how did this come about? And I'll give an example. My colleague, Andrew Bruce, he was a urologist in the 70s, and he looked at two groups of women who get recurrent bladder infections and women who are healthy. And he realized that the E. coli that were causing the bladder infection, they come from the rectum flora, they go to the perineum, they get into the vagina, and then into the bladder. So all the E. coli were there. And he looked at the controls and he said, wait a minute, there are lactobacilli there. They're not in the bladder, but they're in the vagina and the perineum. I wonder if they're protective, right? So his concept was maybe to prevent women getting a recurrent infection, we could flood the area with more lactobacilli. And so that's when I started in 1982, and we started looking at, can you replenish the lactobacilli in women who've had recurrent infections? And really that, for me, was my entry into probiotics. So the concept of probiotics was try and give back some of the beneficial bacteria. And to the extent that we're doing it now, it's somewhat crude because it seems to work, but we're still not basing it on the highest amount of science. And in other words, I can't tell you all the mechanisms. I don't think it should stop us from using probiotics. But when we find mechanisms and when we find that in one woman versus another woman, this works really well and a different one works, then I think the potential to prevent a lot of these infections and perhaps even cure some of the, not bladder, but vaginal and other infections is going to be increased.
How far away are we from finding the mechanisms of action?
So where we are just now, I would say we have a general idea. There are things like, for example, in the vagina, which is primarily where I've worked on, lactic acid. We know that that kills pathogenic, harmful bacteria, and even lactic acid can kill viruses. We know that there are some molecules that are produced that form a surface that prevent the harmful bacteria from coming and sitting on top. We know that there's exclusion, competitive exclusion. Is it absolutely proven? Well, the only way to prove, say, that one specific molecule was key is to do the experiment in a human. We tend to go to animals to do that. I haven't gone that route because I still feel that human is where you need to prove it. And you can have a great experiment in a mouse and it doesn't necessarily tell you that's going to happen in the human. But nevertheless, I think we have a general idea. But as for specific pathways and molecules, I think we're still a ways off, but it's happening very quickly. And I think it's a case of the next 5 years, you're going to see some really exciting science.
What about the idea of prevention versus treatment? You've got probiotics in food, you've got probiotics in medicines. How is that being played out?
Of course, it's controversial because probiotics primarily came out of the food industry. And I think that you'd have to really go back historically where companies, particularly in Asia, France, where they've been used to making fermented foods, and fermented foods like yogurt, for example. And it's a whole different setup in terms of delivery of a product as distinct from a drug company, which would much rather have a molecule and make different parts of the molecule, and they know that every single time they have that formulation, it's exact the same. Well, microbes are living things, and so it's different. And therefore you have food companies that have been in this area, and it means that when you deliver the product, you kind of have to deliver it as a food. That's an issue that maybe needs to be discussed, whether in fact it should be called a food or a drug. And when I go back in history, when all we had was humans, food, water, and air, food was the medicine, right? And in the last 100 years, we've said, no, wait a minute. Foods can't treat, prevent, and cure disease. Only drugs can. I don't agree with that. I think if a food can prevent and treat a disease, we should be able to say that. But that's a regulatory issue, and I think the regulatory system needs to be revamped. And so when you come to the probiotic effects, you tend to look at them not as curing disease, and I'd be the first one to say there's not going to be many cases where probiotics will cure a disease. I mean, that bacterial vaginosis, we've found in one study that it could cure bacterial vaginosis, but it's not going to cure a bladder infection, for example.
Is that severe vaginosis?
Yeah, it's a case of when you have a condition that you can get access for a live organism. So I could see, for example, where you had perhaps a wound condition, maybe it's acne, and I could see that you could put on a probiotic and you could maybe even cure that, right? I see that day coming. Once you get into very serious pathology where it's extremely difficult to access the site, I don't see that happening. And I don't think we do justice to probiotics if we come out and say the primary goal of this is to cure disease. Prevention's a different thing. Prevention can be reducing the risk of something happening. So bladder infections, you might get 7 in 1 year, and you take an antibiotic every day for a year, and it might go down to 2 or 3 a year. Well, that's a reduction from 7. Well, probiotics could do the same.
Alone or in conjunction?
No, on their own.
On their own.
So I would say that's prevention. I would say it's reducing the risk of you getting a recurrence. And in some cases, it may well be just as good as an antibiotic or as another therapy. For example, in colic in children, Lactobacillus reuteri, given as little drops, was shown to be as good as the medicine for colic. And I find that funny in terms of society. So you imagine you've got this baby crying, and the mother and father go out and say, oh, we're going out for dinner, and the babysitter comes over, and mother and father come home and say, the baby's not crying. She says, yes, I gave them some bacteria. They would, oh my goodness, this is terrible, but it worked. What's wrong with that? So in that case, is that treatment? I would say that is treatment of colic and effective treatment. So I think we have to be careful not to go overboard with curing. I think there is a case for prevention, and also there's a case for adjunct. So an example is in H. pylori, stomach ulcers. Probiotics are not going to cure that, but the treatment often includes antibiotics. And if you give the probiotic at the same time, it might lessen the side effects of the drug. Physicians are now suggesting to patients, look, you should take a probiotic when you have an antibiotic or you have something else. And I think that's a good thing. We need to improve the rationale and the thinking, and need to give them guidance on, okay, which ones, for example, H. pylori, which probiotics would be the best to give? And is the purpose reducing the side effects, or is the purpose something else? And that's where the science comes in. Now,
is the science there on that to know the strains? And would it be enough to have a probiotic drink per day, or would it be a higher dose pill? I mean, is that established?
I would say it's not established. I couldn't say to you today, this probiotic A or B has been the proven one, and this is the one I would recommend. And I think it's probably clinical experience would tell some physicians, oh, I've tried this and it worked. So I don't necessarily think there's ones that won't work, but I can't say that we have that information. The other area which we've worked on is on conjoint therapy. So we had 2 papers on treating bacterial vaginosis with the antibiotic and the probiotic. And it wasn't just for preventing side effects and recurrence. In fact, it improved the cure of the disease, which was quite remarkable. And then there was 2 other studies on yeast vaginitis, this time giving an antifungal and the probiotic. Now, I can't tell you we understand the mechanism, but for the patient, it was a significantly improved cure. So I think there will be cases where we need to look at combinations to prevent side effects, and also in some cases to augment what happens. Now, we have done studies in Africa on HIV patients, and anecdotally, we have seen patients who are on very, very toxic antiretrovirals, and they get fatigue and chronic diarrhea, and they've taken probiotic yogurt that we've introduced there. And the reports that we have seen are suggesting they don't have so much diarrhea, they have more energy. Now, I can't tell you that we've proven this in huge randomized trials. We've done small trials. But again, the concept is combining. Probiotics are never going to replace antiretrovirals. Well, not in my lifetime. They might down the road when there's recombinant strains and some really high-tech science. But so we're not, again, suggesting that, but we're saying, look, why not have it as an adjunct to help with quality of life?
In probiotics, there are many strains, and many of them have been researched. And then there are clusters of strains. Is clustering strains of probiotics together an interesting field and one that will grow in the future?
Well, I think what you're getting after with the question is whether or not you can pull different strains together and use them to inform on a strength of evidence on a particular health endpoint. And I think that there is now a compelling body of information for some of the endpoints that we have research on that shows that, in fact, a cross-section of species and strains of commonly studied species, in fact, seem to have similar effects. So, again, if I look back over the 23 years I've been in this field and say, how has the field changed? We certainly, very early on in the field, were adamant about the fact that the effects of probiotics were certainly needed to be considered strain-specific. And I think that there was much evidence back there as it was being generated, especially in animal and laboratory tests, that would certainly show that the effects could be different from one strain versus another strain, even of the same species. But as more and more controlled human trials have been conducted, what we're seeing emerge is that a variety of different strains do seem to have the same clinical endpoint. And so we're probably getting close to the point, if not already there, where we might be able to say for certain endpoints, we might be able to say that it's a common property among a diversity of strains, and therefore cluster the information on those strains together.
So you're mentioning there a point being achieved where there's a compelling level of evidence around certain strains, perhaps certain clusters, certain endpoints. If we look to the US Food and Drug Administration, now their approach to probiotics is one that doesn't please everybody, certainly a lot of academics and people who've worked in the industry for a long time, like yourself. What effect is their approach to probiotics having on the market and on research as well?
I'm actually quite worried and concerned about the FDA and their stance about probiotic research. The reality of it is that regulatory bodies tend to think of things in boxes. And in the case of the FDA, they have a box that foods fit in and a box that drugs fit in. And the food box also contains kind of another smaller box, which are dietary supplements in the US. But there's drugs and then there's foods. And the FDA's reading of the Food, Drug, and Cosmetic Act is that basically all effects in humans are drug effects. And the only effects in humans that you can study for foods include taste, aroma, and a very narrow definition of nutritive value. Now, this stance is being reviewed right now, and it may change in the future. I can hope it will, but I fear that if it doesn't change, that essentially none of the research that needs to be done on probiotics on these human health endpoints will be able to be conducted in the United States. It will be the end of that research as we know it.
And you're seeing that effect already?
Well, probiotics have had the unfortunate situation where they've been reviewed by the FDA and many of the investigators, even though they have either private funding for their studies or NIH funding for their studies, and the IRBs, the institutional review boards that look at the safety of studies, have no concerns about the studies. The studies are still being told, or the investigators for these studies are still being told by the FDA that they need to conduct these studies under the drug rubric, which means that they have to apply for an investigational new drug in order to be able to do a study. And this is true even when the product being tested is a product that essentially can be pulled off the grocery store shelf and you could eat on the way home, from the doctor when you've got your antibiotic prescription, and you could be eating it in the car on your way home, but the FDA views that use as a drug use, and therefore it can't be researched as a food, it has to be researched as a drug. And that has very negative implications about the progress of research. Possibly the biggest concern with that, and there are many, is that the FDA seems to impose safety studies, which can be quite expensive and quite unnecessary for certain types of studies. But if you're conducting a drug study, the very first study you do, has to be a safety study, and it has to be a safety study in adults. It can't be one in children. So rather than doing much needed research that talks about the effectiveness of a probiotic to have certain biological effects, instead we're doing safety research that none of the scientific community thinks is necessary.
Yeah, and again, I'm taking the research from health or optimizing health or maintaining health towards disease.
Well, in this particular case, the FDA doesn't really care if you're optimizing health or going to disease. They're considering it all drug research, and that's part of the problem. Foods should be able to promote health, and those studies should be able to go forward in humans. The drug research is drug research, and that has to be done under an IND, but it's just that the FDA seems to have a very broad interpretation of what constitutes drug research, and they're incorporating much of what we might think should be food research.
Do you see any change in that system in the near future?
The FDA's process is under review. They are accepting comments on the process right now, so we'll have to see if they decide to make any loosening of that approach or not.
And at the same time, companies can make structure-function claims, food companies.
Yes, so what you're hitting on here is the distinction between regulatory barriers that might exist for conducting research in the United States versus regulatory barriers that might exist for making claims. And in the United States, we actually are able to have claims on foods that are of the nature of what you just mentioned, the structure-function claims. And what a structure-function claim is the relationship of the food to the normal structure and function of the human body. So it's not a cure, treat, prevent, or mitigate disease claim. It's relating to the normal function of a healthy body. And an example of that would be supports a healthy digestive system. So those types of claims are allowable on foods and dietary supplements in this country without FDA approval. And so you'll see quite a few of those claims being made on products. The problem we're having now is that going forward, it won't be possible for companies to conduct the research to substantiate those claims in humans in the United States.
Let's go to a basic question. What is a probiotic?
A probiotic was defined in 2001 by a consensus panel that was convened by the Food and Agricultural Organization of the World Health Organization. And that definition that was adopted back then was that probiotics are live microorganisms which, when administered in adequate amounts, confer a health benefit on the host. And that definition has prevailed, I think, through the years, and here we are, 13 years later, and the definition, I think, is still very broadly used. Many regulatory agencies have accepted it, and most scientific organizations have accepted it, and I think it's a very functional, broad definition that encompasses the core of what a probiotic is.
What about in terms of the way that the food industry applies probiotics in the terms of the strains they select, the formulations they make, the way they market the products? Do you feel like the food industry is doing a good job with probiotics?
The food companies are in a tough position. I give them full marks for the fact that they have done research. These are companies that don't have the sort of return on investment, say, that the drug companies have. They can't charge the prices that they charge. But some of them have really gone out of their way and have done excellent R&D. Sadly, that has been stopped or reduced in the last few years because of legislation, particularly in Europe, where they've stopped allowing a claim. So a company in Europe can't even write the word probiotic. Well, I shouldn't say Europe, I should say the EU. They can't write the word probiotic. So in Ireland, if you buy a probiotic yogurt, it can't say probiotic on it.
Or prebiotic.
No, that to me is ridiculous. But the food companies, they've done a great job. They've, I think, got wonderful ways to deliver these organisms. Because people love to take food. They love food, certainly in Europe. Maybe North America, we're a bit more used to drugs and pills and tablets. But so I think they've done a good job. But they're limited. And I think this is a great time to help them out. And by that, I think there's a few things that could be done. For example, in Canada and the US, why wouldn't we have a separate funding agency that just looks at probiotics and the microbiome? And you say, well, why not? Because what we're trying to do is we're fitting it into infection and immunity and metabolism and diabetes and all these other sections of NIH or sections in Canada. This crosses everything. It goes from food to natural products to medical devices, even as far as drugs. So this is a great opportunity to help the food companies and help develop our understanding of what they do and how we can deliver it. And, you know, we're only at the cusp, because the microbiome has now identified organisms that look to be really helpful for our health. And under the current system, they will never be in a food because there's no safe history of them being in a food. So they will be developed as drugs. So patients will have to wait 10 to 15 years for something that is already part of the system, could help them probably quite quickly, potentially could be put in a food, but there's no way they'd be allowed. So it's going to be a challenge for society, and by that I mean the regulatory society, the food companies, and the drug companies are sitting here because they've been used to a nice little molecule, as I said earlier. Well, this isn't a molecule. And you have to change the whole environment in the company, because the people that have worked there have been used to, say, chemistry or engineering and depending on the industry. And now they've got to be into fermentation and microbiology in a different way, and it's maybe not patentable the same way, it's not delivered the same way, it might require a different handling rather than room temperature pills that last for 2 years. So you're going to have to change the culture within the pharmaceutical industry. And you might say, well, that's never going to happen. It's already happening. And pharmaceutical companies have realized, wait a minute, there's amazing potential here, because not only are we struggling with drugs that we've been in the business for 50 years and finding the new generation, especially in antibiotics, but they're also finding, just a minute, some of the molecules in these organisms might be the key, and that could be developed as a drug. So I'm finding that the food companies, I think, need help and encouragement. The drug companies, I would say, need guidance, and together they could work to create some really, really terrific new interventions.
There's the gut and there's the brain. Are they linked?
Amazingly, they are, not just by the bloodstream and the lymphatic system.
There's been some lovely work done, particularly in Hamilton in Canada and some in Sweden and the US, where they've realized that the bowel, which is down here, the small bowel intestine, is packed and packed with bacteria. And the miracle of humanity is that those bacteria could kill us if they got access to the bloodstream, just because there's so many. And that's why with appendicitis you worry about the bursting of the appendix, why if you have a wound you worry about the intestine bleeding. Well, there's one layer of epithelial cells that protect humans. So the bacteria in the stool, so in the food, that are moving through the bowel, and all these bacteria, there's one layer of epithelial cells. And if these epithelial cells are damaged, and if they're loose, then you can imagine the bacteria can get across that epithelial barrier. Well, that's fine, but what about a healthy person? Well, it turns out that the bacteria are communicating with the host, and they produce these molecules. We call them neurochemicals. And these neurochemicals, what do? Well, they're signaling the brain. And they've shown that a lot of this goes through the vagus nerve, so it comes up through the nerve into the brain. And you say, okay, what does it influence? So they've done these really neat experiments. I love to show them again to students. And I say, what would happen if a mouse came in the room? Where would it go? Well, it would go around the outside. And it's scared to go in the middle because it feels vulnerable. So that's a stress response. And these researchers did a study, and they gave them probiotics, and it found that the mouse spent more time in the middle. In other words, its stress came down. So if it affects stress, wow, that's interesting. Now, what about memory? And it looks like it might have an effect on that. So then you start to think, okay, these are pretty important brain functions. And I show students the fetus. And you look at the fetus from week 3, look right through to week 40. And there's particular times when we know the brain develops. Well, what are we doing to influence that development? We give iron and folic acid. Should the mother have certain bacteria in the gut that influence neurochemicals and therefore the development of the brain in the child? Wow, wouldn't that be exciting? So this takes us on to, okay, can you radically change the microbiome? And how might this impact not just brain but other functions? And fecal transplant is kind of come into being a very important concept. And it started with people having Clostridium difficile. So the best way to, simplest way to explain it is you get lots of antibiotics. It kills a lot of the organisms in the gut. And this organism called Clostridium starts to multiply, produces a toxin, and sadly it kills people. And for example, my wife's grandmother went into hospital with a broken hip. 2 days later, she had C. diff and she died. And in Quebec and Canada, there's been thousands of deaths and a lot of cases. So this is really difficult, and chronic C. diff infection, as they call it, is a terrible thing to have. So people decided, okay, why don't we take someone else's good stool, bacteria, half of what we poop is bacteria, they say. Why don't we take it and put it into the person that is really in trouble? Well, they thought, this is crazy, we'll do it. They did it, it cured it. 90% cure rate. Oh, this is incredible. What does that mean? Well, how do you do it? It's very simple. You take the stool, you mash it up. I know this sounds awful. You take the liquid, you put it in a tube into the rectum, or you put it down in Holland. They've done experiments where they put it down and it goes right into small intestine and it cures the disease. Unbelievable. So that's called fecal microbial transplant.
Is that being used in the medical? Is that it's just in experimental?
No. It's been used in hospitals. In fact, there's even guidelines on how you can do it yourself, and I don't suggest it. And the really important part of this, if you're taking someone else's stool, you have to make sure they don't have HIV, hepatitis, they haven't got an inflammatory condition or cancer. So there has to be very stringent analysis of the stool. But going back to your point about the gut-brain, well, what happens if the organisms that you put in could have a beneficial effect on the brain? Because remember I said they're producing these neurochemicals. And people are now thinking, well, it's maybe too much of a stretch to look at dementia and Alzheimer's and Parkinson's and other conditions, but maybe there are stages in those conditions early on before there's real pathology that fecal transplant could have an effect. And so this has now opened up a whole other concept. It's by far, it's not proven at all. Clostridium difficile is proven. It's also made people worry, well, if you have a donor of stool, what did they have for breakfast? And do you need to have them on a specific diet? And what's the long-term impact? Yes, it cured C. diff, but 10 years down the road, are they going to develop some other disease? So lots and lots of questions have come from it. But the concept is really, really cool.
Well, I hope you enjoyed all these wonderful insights into the cutting edge of microbiome and probiotic research. The science continues and the potential is immense. Stay tuned for more developments. I'm Shane Starling.